time for linkage practice. So we talked about linked genes and how one piece of chromosome can have multiple genes upon it. And that means that they are linked together. We call them linked. So here we're looking at pea plants. So we're looking at 4 different traits: height, flower color, seed shape, and pod color. These are all on the same chromosome. Okay, these 4 traits are linked on the same chromosome. Crosses were performed in which linkage of the gene was studied. Use the following to determine the relative mapping of this chromosome. So what I want to know is, on our chromosome, what order do these genes appear in? So this is our trait. This is the alleles available for that trait. So again, we have here, here, and here. So I'm going to call the gene for these. This one is going to be gene H. This one's going to be gene F, gene S, and gene P. I want to know on this chromosome, what order are they? Is it H, F, S, P, or is it something else? What order do they appear in? To do that, we need to see what the relative distance is from each of these genes. If I look here, I went from this to this, so that would be my height. So this is looking at from height to, and our B's are flower color. So we're looking at from height to flower color. I crossed 2 parents heterozygotes for each trait, and these are the results I got. First thing I'm going to do is I'm going to find my total. When I add all of these together, my total is 1,354. Now to find our frequency of recombination, I use my recombinant types. So these are tall and blue. Okay, and they would also give me short and white. My recombinants are going to be the lowest frequency ones here. So that's where crossing over happened. It gave me these. And if you remember back to previous lesson, to find our frequency, I take my recombinants, divide by the total, times 100. So my recombinants, when I add these together, I'm going to get 91 divided by my total times 100. So I know that the recombinant frequency for this is 6.7%. I look at the next one, and from here I'm looking at height, the distance from height to, and our S's were seed shape. So height to seed shape. I can add them up and find my total, 1,433. I can look at my recombinants. Those are the 2 smallest values. The total for my recombinants is 299. I divide that by 1,433 times 100. Okay, and that is going to give me, again, 6.7%. I go to the next one. Here I'm looking at height to pod color. So height to pod color. I then can find my total is 1,770. I can find my recombinants. That's going to be 359 divided by that 1,770 times 100, and that will give me 20.1%. The next one here, I have from flower color to seed shape. My total is 2,000 259. My recombinants, my two lowest ones, is going to be 371 divided by that 2,259 times 100 will give me 16.4%. And then for my last one, I add these up. 1,460. I add these. I get 360 divided by 1,460 times 100 equals 24.7%. And I'm sorry, I forgot to put this in here. This is from flower color to pod color. Now I have my percent recombinant, okay, or a frequency of recombinants, and I can use that information to create my map. So I'm just going to write down my different recombinant distances. We have H to F equals 6.7. H to S equals 6.7. H to P equals 20.1. F to S equals 16.4. F to P equals 24.7. Now that I have those, I'm going to start creating my map. To create our map, we start with whatever has the greatest distance between the two. So I'm going to start down here. I'm going to say that from F to P is 24.7. Excuse me. Now I can start looking at what do they have in common. Well, I have F to H and H to P. So I can look at those. F to H is 6.7. So that means H is either over here or over here. But I'm not sure which. But if I look at H to P is 20. So from P, I can go back to H. And if the H is here, that would about work. If H was over here, wouldn't work. If H was over here, it wouldn't work. So we're going to put our H right here. We're going to say that F to H is 6.7 and H to P is 20.1. Now, if I add these together, I get 26.8, which is bigger than this. But that's okay because I'm looking for relative distance. Okay, and you have to think that if I'm looking at the very edge of H, and maybe H takes up this much space. So it could make things different. Okay, so we're looking for relative, and it doesn't have to be exact. But that means I took care of this one and this one. All I have left is H to S and S to F. So S to F is 16.4. That means that a S could be either over here, or it could be way back over here somewhere. But H to S is 6.7. Now, H to S, if it was over this way or over this way, so this one over here, it wouldn't work. It'd have to be this one. So S is going to be here. And from H to S, this distance is 6.7. And from F to S, that distance is 16.4. Okay, so I've discovered that my relative mapping is F H, S, P. Make them a little more equal distant there. So I know on this chromosome, this is the order that these genes are found. Okay, that is the answer. Okay, now let's take a look at number 2. Assume the genes are not found on sex chromosomes. That means it's not on an X chromosome, so we don't have to worry about male versus female. Indicate the body color alleles for brown, black, and wing size for normal wingless. So we're looking at fruit flies, and I know that because we have wings, and that's what we've been talking about. All right. The genotype of flies in the P generation are, so it says we're crossing. Okay, that means that we're going to use purebred of one and purebred of the other. It doesn't really matter which one we use for male and female. However, traditionally, we would use purebred dominant for the female and purebred recessive for the male. Now remember, the P generation are purebred. If I look at this and we're to do a cross, the only gametes I can make here are capital B with a capital N, and the only gametes I can make here are little b with a little n. All of the offspring would be heterozygous for both traits. So the genotypes in the F1 generation are like this. Now, a female in the F1, so one of these, crossed with a true breeding black wingless. So black wingless. And I know that because black was recessive and wingless is recessive. Okay, so I'm now going to cross this and this. When the flies are mated, you count offspring of 1,600 offspring in the F2 generation. If the body color and wing type genes are not linked, how many flies of each phenotype would you expect? Okay, that means that we're going to just do a regular old cross. So if I were crossing these two individuals, I'm just going to slide up a little so you can see. Okay, if I were crossing these two individuals, I can look here in my gametes. We use foil. And for this one, I can do foil, but they're all going to be the same. So I only really need to do one row. And when I bring them back together, this is what I would get. Now, when I look inside, each box is different. So that means I would have a ratio of 1 to 1 to 1 to 1. That means I should have equal numbers of each. It tells me that I have 1,600. And I have 4 different kinds. So that would mean that of those, I would expect to have 400, 400, 400, 400. Number of brown-winged flies, okay, from above. So we know the number for each of these is going to be 400 because they're all 400. Brown and winged. Okay, when we look up above, brown is dominant, black is recessive, normal is dominant, wingless is recessive. So I need something that is dominant with dominant. So when I look here, that's this one. Black with wings. So that would be 2 recessives. We have to have at least 1 dominant for the wings. Black but wingless. So that means recessive, recessive. Brown, so that means I have to have at least one dominant. And wingless, 2 recessives. So that means we have this. So my expected would be 400 of each. And it says, whoa, where'd we go? There it is. When you count the F2 generation, you actually got 85 that are brown winged, 728 black winged, 712 brown wingless, and 75 black wingless. These results indicate that the body color and wing size are linked genes. Okay. What are the phenotypes of the recombinant flies? Refer back to the P generation of the first paragraph if you need to. Okay. Calculate the recombination frequency between the linked genes for body color and wing size. That means that we want to go back to figuring out our frequency. So we know that we have 85 brown winged, 728 black winged, 712 brown wingless, and 75 black wingless. We can look back at the first part where it talked about our P generation. And the P generation, we have brown winged and black wingless. So those we know are the parental. We want the ones that are a combination. So that would mean black wingless or brown winged. And we know our recombinants are always the low, low, low numbers. So those are our recombinants. To find our frequency of recombination, we do like we've done before. We need to find our total. Grab my calculator here, get my camera to refocus a little bit. So I add these up. 85 plus 728 plus 712 plus 75. 1,600. And that matches what they told us. They told us 1,600. So we know we've got the right numbers written. Now, to find our frequency, we take the recombinants divided by the total times 100. So I take the total of my recombinants, which is 85 plus 75. That's 160. So we get 160 divided by 1,600. Okay. Now, that times 100 gives me 10%. So our recombination frequency is 10%. That means the distance from color, to wings is 10 map units. And we get 10% recombination. That's the end of that practice sheet. I'll have some more videos for you soon.